Hello and welcome to another episode of the Omics cast. My name is Alberto Veleda and we're going to talk about single-cell omics of centenarians. The paper I'm going to talk about is a preprint on bioRxiv entitled Single-Cell Transcriptomics Reveals Expansion of Cytotoxic CD4 T cells in Supercentenarians. And it's a paper from Japan. The last author is Piero Carninci, which is someone that I follow on Twitter. I find him very interesting. I recommend you to follow him as well. And in this preprint, as you can see, it's a preprint with the pre-formatted way of submission. They talk about supercentenarians, which are people that have reached 110 years of age compared to centenarians for people that have reached 100 years. And they do an omics analysis, a single-cell omics analysis, to find out what may be happening to them that predisposes them to live for longer than average. And what they do is they perform a single-cell transcriptome analysis of peripheral blood cells for 7 supercentenarians and 5 younger controls. So this is a small data set, but as we will see, they found quite a lot of biology behind. And what they found is an increase in the CD4 CTLs, which is a type of blood cell, that's coupled with a substantial reduction of B cells, a different type of cell, as a novel signature of supercentenarians. And so what they argue here is that supercentenarians have something going on in their genetic makeup or in their epigenetic state that makes them live longer and do so by having a better immune system at their age than the average population. And they also found out something that biologically is quite relevant because it's something that's been investigated heavily recently, which is that they find CD4 CTLs massive clonally expanded in their bloodstream, which is something CHIP has been found recently. Clonal expansions of cell lines, something that happens sometimes in cases like cancer or so. And so here they found it in a positive case, which is for people that live for a long time. And so they argue that supercentenarians may have unique characteristics in their circulating lymphocytes that allow them to sustain immune responses to infections and diseases. They describe what supercentenarians are, which are people that live for more than 110 years. And they say that they're very rare, and there are only 100 of them in countries like Japan. And they say in their life they have a long lifespan, and also they maintain relatively high cognitive functions and physical independence even after 100 years of age. So it's not only that they live for longer, they also have a better quality of life at points in their age where other people have already started deteriorating. And so they say there must be a reason why these people can spend most of their entire lives in good health, and they go and investigate it. They say many functions of the immune system show a progressive decline with age, a phenomenon that's known as immunosenescence. Senescence, like when a plant starts to wilt. Here, a human being starts to wilt, or their immune system starts to wilt, and it leads to a higher risk of infection, cancer, autoimmune diseases. Whereas these people that age so well, they show a low level of inflammation, and that is the best predictor of successful aging. So what science did they do? They profiled PBMCs, blood cells, from 7 supercentenarians, SC1 to SC7, and 5 controls, CT1 to CT5, aged 50 to 80. And they used droplet-based single-cell RNA sequencing from 10x Genomics, the standard method that 10x Genomics sells for this, to do single-cell RNA-seq, to find out what populations of cells are in this people's blood and what characteristics they have. And they did this on a median or mean of 4 to 5 or 6,000 cells per individual, as it's shown here. And what they do, they essentially fact sort these cells. So they do a sorting of a specific subset of cells, and then they analyze these cells through the single cell library prep of 10x genomics, right? So for 7 supercentenarians, 5 individuals, they take the whole blood, isolate the PBMCs, and then they produce the libraries, do the sequencing, and then analyze via gene expression profiling and visualize in the cloud plots. This is an example of the cloud plot here, which is colored by the presumed types of cells inside. And so they recover about 4 to 6 to 8,000 cells per individual, and then they produce clouds like this, with the different populations here colored based on the prediction of what they are. T cell population 1, T cell population 2, B cell, natural killer cells, etc., etc. And so what they find is that some of these populations change dramatically between individuals, and that this change in the populations, in the composite of these different clouds, different colors, is an indication that something biologically is happening differently between supercentenarians and the rest of the population. And another thing that they found out is that the progression of some of these clones changes radically a long time, right? And so this is a way of plotting in where basically you look at different clones of cells and using something called the pseudo time estimation, try and find out how these clones evolve in time from the different types. And so they conclude that some of these supercentenarians, they have this clonal expansion of some of these CD4 populations, and that is something that is probably helping them have an immune system that's in a better shape than the rest of the individuals, the 50 to 80-year-old individuals. So not only is an increase in the population, but also a reduction of another type. And how did they validate this? You can say, well, we've done all these single cell omics. How do we believe that all this is correct? Well, they validated the same phenomenon they saw in the different color clouds by fact sorting B cells and looking at their relative quantitative amount in supercentenarians to controls. So they did 4 more supercentenarians and 3 healthy or younger controls, and found that what they see in the single cell omics analysis, they can also see by box standard FACS analysis. A little bit on the methods. The Omics cast is about methods as well, apart from the biology you find in them. And so a little bit about the methods that they applied. They applied the 10x genomics RNA-seq method that produces these GEMs, the gel beading emulsions. Then they are all bar-coded and PCR amplified and then sequenced. The sequencing was done on Perrin Illumina HiSeq 2500 machines, mostly 1 sample per lane. So if they do 5 to 6 or so thousand cells per individual, then they put 1 in each lane, right? So this will cost you in the high hundreds or low thousands of dollars per sample, and they did about 12 samples. Then they applied the software that comes with the 10x Genomics kit, which is Cell Ranger, and then they did the FASTQ generator, the counting, etc., and then they mapped all at GRCH38. And then after that, for the analysis of the T cell subsets, they use an R package called SURAT, which was used to analyze the T cell subsets. And they use different functions in this SURAT package. Here I highlighted some of them. Find clusters, run TSNE that produces the plots, find markers, TSNE plots, etc. And then they did this study on how the clones evolve along time. And they do this with a piece of software called Monocle. And this Monocle software estimates a pseudo-temporal path of T cell differentiation. They basically say, if some of the T cells are in a naive state or an early state, and some of them are in a mature state, let's find by the population of cells we have this path along the pseudo-temporal space. And so these pieces of software were applied to the data set that we described to get the results that we've shown. Now, what are the conclusions of this? The conclusion is that single-cell omics is a very high-resolution method to understand biology, to understand the biology of a population of cells, in this case in the bloodstream, including the biology about the immune response of cells or the immune state of an individual in their bloodstream. T cells being a very important part of that. It enables the discovery of projects to catalog what is seen, right? So this is a cataloging project or a discovery project, if you want. And then we can come up with hypotheses of why this happens by linking it to previously known science, right? People knew about populations in blood cells changing along age. They've done the fact sorting here of the B cells, and they saw that they decrease in time. So what we can do with methods like single cell omics is generate these reference data sets in discovery mode, find out if our previous hypotheses match what we see in the results, but only that once we have the results, link it to other hypotheses to do further studies. And so what's the malarkey with looking at centenarians or supercentenarians? Why are we looking at these old people that are going to be dying anytime soon? Well, these people live longer, which means we can develop and tailor treatments for the entire population based on their genetic makeup and epigenetic status. Meaning, if we know what genetic makeup and epigenetic status is found in these centenarians or supercentenarians, if we know from these epigenetic marks or their genetic state makeup, what is that makes them healthier, in this case by looking at the immune system in their bloodstream, we can then say, okay, if we replicate the same thing that's happened biologically to the centenarians, supercentenarians, if we replicate that by interfering with the state of a normal individual and bring it to the similar state where these centenarians are, we are tailoring this therapy to the genetic makeup or epigenetic status of the individuals. And this is essentially the premise of precision medicine. So this is a good example of applying omics technologies, NGS, to precision medicine. Thank you for listening to this episode. There's a bunch more on the website, and if you want to know more about them, you can follow me on twitter.com/Albert Vilella. And you can also follow me on LinkedIn if you want and connect with me. I'm happy to reply to any questions you have if I have the answer. And also a reminder that this slide deck is available upon request. Until the next episode, goodbye.